Hi, everybody. Hopefully you can hear me. I am Sarah Ireland, the Chief of Communications and Growth here at the Great Barrier Reef Foundation. And I just want to thank everybody for joining us here for this deep dive webinar. So whether you're tuning in from Australia or somewhere else across the world, thank you for being here. I'd like to start by acknowledging the traditional custodians of the lands on which I'm on today in Meanjin, in Brisbane, the lands on the Turrbal and Jagera people. And I'd like to extend my deepest respect and recognition to all traditional owners across the country who are connected through land, waters and sea country, including across the Great Barrier Reef and its catchments, and pay my respects to elders past and emerging. Now, to set the scene for today, first of all, I just want to take a step back and look at the bigger picture. Where is the reef today and the role innovation is already playing in shaping its future? Many of you, particularly those here in Queensland, would have seen the recent coverage of Cyclone Narelle hitting the North Queensland coast. And while the impact of that event is still being assessed, one thing we know for certain is that events like this are becoming more frequent and more intense. And they're part of a broader pattern of pressure on the reef. So from rising ocean temperatures to increasing severe weather events, it's no secret that the Great Barrier Reef and coral reefs globally are under real pressure. And for the first time in history, coral reefs have also reached a global tipping point, meaning that their damage is becoming harder to reverse. But as those of you who do follow the Great Barrier Reef Foundation across our socials, receive our newsletters, you'll know one of the things we continue to say is that there is still time to act. Across the reef, a wave of emerging innovations is offering new hope. Practical, science-led solutions are already being tested, refined, and scaled in the water today. So before we dive into today's discussion, so firstly, I do want to say if you have any questions throughout any of the webinar, please do pop them in the chat and we'll find some time at the end of the session to answer some of those questions. Those we don't get to today, we'll make sure that they come out in our next newsletter. And I would like to kick us off by playing a short video to give you a closer look at some of the challenges facing the reef, as well as the opportunity we have to respond.
People think of coral reefs as a great place to visit and to have a photo of and swim amongst. But fundamentally, they're a home. They're a home and a critical habitat for a quarter of all marine life on the planet, and we are a blue planet. So if we lose coral reefs, it is a domino effect of ecosystem failure. There is no doubt that the number one threat to coral reefs is climate change. People knew that coral reefs were fragile, more fragile than people had expected, and that they were at the front line of a changing climate. What we're seeing on coral reefs right now is a wake-up call and a cry for help, that oceans are not able to continue to just absorb all these pressures. They need the pressures to be reduced, but they also need us to dive in and restore these ecosystems back to their former glory. And along the way, we're trying to find a way for those coral babies to be tougher, to adapt to future conditions so that they'll survive future marine heat waves and future events. We know so much more about how coral reefs tick. We know how to rebuild coral reefs at scale, and we're really getting close to being able to put these promising prototypes in the water. So we're organised, we're smart and we're determined. And I guess in the privileged position I have in the Great Barrier Reef Foundation, I see what's coming down the pathway. So I am hopeful, but I also know that we must be tenacious and determined if we are to be successful this decade.
So as Anna touched on in that video, while the challenges facing the reef are significant, there's also this really incredible innovation that's happening right now to help protect and restore it. So today we're going to have a closer look at some of those solutions in action and hear directly from some of the team members who are leading this work. So first of all, I'd like to welcome Dr. Mary Bonin, who is working at the forefront of reef innovation, improving the way that we detect and respond to crown-of-thorns starfish outbreaks before they cause widespread damage. And we saw some crown-of-thorns starfish in that video as well. We'll show you a little clip soon, so you'll be able to see them a little bit more. So Mary, welcome. Thank you for joining us.
Hi.
Maybe you could tell us a little bit more about your role here at the Great Barrier Reef Foundation.
Sure. Thanks, Sarah. Welcome, everyone. It's really great to be here, and thank you all for joining us. I'm the director of our crown-of-thorns starfish control innovation program. And in this role, I'm lucky to work with a whole bunch of really brilliant scientists from across Australia who are developing innovations that help the people that are working on the front lines to protect the Great Barrier Reef from these massive starfish outbreaks.
Brilliant, thank you. So some people think of, we see starfish every single time we go to the aquarium. It's on kids' cartoons. We see these like cute starfish. People think they're harmless, but crown-of-thorns starfish, they're very different, and especially when they congregate in large numbers. Can you explain what they are so people really understand what we mean when we talk about crown-of-thorns starfish? There we go. And what kind of damage outbreaks they can cause on the reef?
Yeah, so these are not your typical cute starfish. They are, as you see on the vision there, spiky, venomous, and really coral-eating machines. That's what they do best. So they're native to the Great Barrier Reef and reefs around the Indo-Pacific, but they have this tendency to have population outbreaks and booms and busts. And that kind of like a locust plague on land, you can imagine. So when their numbers outbreak, they can be present in tens of thousands on a single reef and do a huge amount of damage, eating 90% of the live coral that's there. And then they can also spread to lots of reefs, so they can impact large areas.
Yeah, so 90%, so they can have that huge damage. And I think I've heard before a dinner plate size, they can eat a dinner plate size coral per day. Is that right?
Every single one can eat that. And so when you multiply that by tens of thousands, you can imagine how much damage they can do in a relatively short amount of time. And on the Great Barrier Reef, we've seen 4 recorded outbreak waves since the 1960s, and we now know the 5th outbreak wave has started. And these outbreaks historically have caused huge amounts of coral decline, 40% of the coral decline on the Great Barrier Reef. So that's why Australia has a control program that operates across the reef with 6 vessels, over 100 crew out on the reef every day culling the starfish. And we know these crews can be really effective, with starfish numbers 6 times lower and coral cover 40% higher when they get in early and get to work. And that makes a lot of sense. You can think about pest outbreaks. You want to be on the front foot managing them rather than the back foot.
Yeah, absolutely. So we've got these teams that are out there, and we've got the programs in place. So can you talk us through how outbreaks are currently detected? And both, I guess what's great about those, that we are looking at them and we are trying to detect the outbreaks, but talk me through some of the limitations our current methods have.
Yeah, so you can imagine one of the biggest challenges to controlling these outbreaks is that we're trying to do it across a huge area. The Great Barrier Reef is a huge place. And so we're searching for those early signs of the outbreaks. But our main detection method right now is a pretty blunt tool for that purpose. It's called a mantitow. And what actually happens is it's a diver-based visual method where a diver is literally holding onto a rope and towed behind a boat, looking for signs of the starfish. So it's really manual. And imagine doing that across the 2,000-kilometer length of the Great Barrier Reef. It takes a long time. So the current method isn't particularly scalable, and it's also not very sensitive. So it kind of tells you when you've already got a problem, not when the problem is coming.
Yeah, and I can also imagine if you've got someone just being, underwater. I've seen a video of it. It's like, for those of you who haven't, it's basically holding what looks like a kickboard underneath and being dragged behind. And I can imagine, Mary, in some parts of the reef, that's also not very safe for the diver themselves.
People joke about being big human lures for whatever's in the water. So safety is also another thing we're mindful of.
Yeah, okay. So with that in mind, then obviously we're here to discuss the innovations driving recovery across the reef. So what is one of the new tools, and I know you've been part of the development of a new tool to detect the outbreaks. Can you talk us through that?
Yeah, so let's talk about environmental DNA, or eDNA for short. So just like us, we all shed little bits of skin tissue, hair. Starfish do the same thing. So out in the and that little fragments of DNA actually are free floating in the environment, and that's what eDNA means. So together with our partners at the Australian Institute of Marine Science, we've developed a genetic tool that actually detects those tiny fragments of starfish DNA floating out in the ocean, which is pretty amazing. And the way that works is that the people out on the water, on a vessel, on a tourism pontoon, can collect DNA samples by pumping seawater through little filters. And that's now a really quick and easy process. That's something we've worked to refine. And then those samples get sent back to the lab to get analyzed. That whole process takes about 2 weeks, and then the results are provided to the control teams to help them prioritize when and where they need to do the culling.
Yeah, amazing. So what makes this such a game changer then?
Yeah, well, I mean, this is really world-leading stuff because it's a really sensitive tool. So we can actually quantify the amount of starfish DNA in the water with this method. So it goes beyond telling us just presence, absence. Obviously, these are a native species. We expect them to be present, but what we need is actionable information about when numbers are increasing and starting to reach those levels that cause concern. So this really is the first early warning tool we have to give us a head start before those starfish start to build to levels that cause coral decline. So we really can't do that with the current method. And of course, as you mentioned, the other thing is that you don't even need to get in the water to do this. So it's safe for people to do, who don't have dive training, and it's safe to deploy even when conditions, whether there's dangerous animals or conditions in the water aren't good for actually getting in the water. So it's really, has lots of benefits, and it was this method that really helped give us that early warning to the 5th outbreak wave building on the Great Barrier Reef. So that allowed our partners at the Great Barrier Reef Marine Park Authority to deploy control crews to start that proactive pest management for the first time in the history of managing these outbreaks.
Yeah, wow, that's amazing. So you mentioned right at the start that what you do at the foundation as part of the crown-of-thorns starfish control innovation program. So I guess the key is in the name there, the control innovation program. But as part of that program, what are you hoping to test or prove next?
Yeah, I mean, well, there's lots of exciting stuff in the pipeline. One of the things that I'm excited about with the eDNA method, I'm a really practical person, and as you can probably imagine, cutting edge tools like this aren't cheap. So right now our partners are working to develop new lab techniques that bring the costs down. So this is more cost effective. If it works, it'll reduce the lab costs by 25%. So lowering costs is always a good thing, because really the point here is that this isn't just innovation for innovation's sake. You know, we're really about developing tools that have practical applications, are feasible and cost effective for the people who need them.
Yeah, so we're really using this to, I mean, we talk about this at the foundation in our reef, our other reef restoration work, but making it, making the work faster, making it more cost effective or cheaper, and looking at how we can kind of scale it across the huge expanse of the Great Barrier Reef. So
absolutely.
That's brilliant. So last question, before we'll bring you back at the end for any other Q&A, but what gives you optimism? Obviously, you're doing all this amazing work, looking at how we can overcome all these different challenges, but where are you most hopeful about this work and its potential to protect the reef?
Yeah, I mean, I've been studying coral reefs for 20 years now, and they are really pure magic. I mean, I've seen amazing things, and I've also seen them hit by some significant challenges, these starfish outbreaks, mass coral bleaching, and I've witnessed their ability to bounce back if they're given a chance. And these kinds of tools that we're talking about today are the things that give that reef a chance to bounce back. So that absolutely gives me hope. And I think the other part of this, Sarah, is that, the work we've been talking about today is part of this innovation program, which is a really collaborative effort with a lot of brilliant and passionate people who are, all focused on meaningful solutions to what are complex problems. So that collective action and, shout out to everyone here today. All the interested people here today, are what give me hope. So.
Wonderful. Thank you, Mary. We really appreciate your time. And that was incredibly insightful to just hear a little bit more about how we better detect and respond to threats, particularly crown-of-thorns starfish on the reef. So thank you.
Thanks, Sarah.
So
thanks, everyone.
See you later. We'll see you in about 15 minutes. But now we will look at how we're working to future-proof coral, strengthening its resilience in a changing ocean. So I am joined by Dr. Petra Lundgren, who is going to share some insights into cutting-edge research, exploring how we can strengthen coral's natural ability to cope with warming ocean temperatures. Thank you, Petra, for joining us.
Well, thank you, Sarah, and thank you, everybody, for joining us in this kind of really exciting webinar. As Sarah said, I am also working here at the Foundation. I'm the director for the coral restoration programs. And I do kind of come from a background. I've got a PhD in coral genetics. I did my postdoctoral research on the genetics of heat tolerance in coral. So this is really a bit of a passion project for me. So super excited to be here to talk to you all about this.
Wonderful. Thank you. So, we'll jump right in then. Can you help us understand what happens to corals when they're exposed to heat stress? So we talk a lot about a bleaching event, but what does that actually mean? What's actually happening during a bleaching event?
Yeah, so corals live in symbiosis with this tiny, tiny little algae that lives inside its cells, and it provides a lot of the nutrient requirements, like a lot of the food for the coral. And it gets that through photosynthesis, which is pretty much how plants create sugar and oxygen for their survival and nutrition. And during extreme heat, and especially like intense sunlight scenarios, this photosynthesis goes into overdrive, and the oxygen side of that equation, in a very simple term, gets a bit toxic for the coral host. And as a result, it spits the algae out. You can't be here anymore. You're really uncomfortable. You're behaving really weirdly. I need to get rid of you or I'm going to die. And that's when we say the coral is bleached, because all of a sudden, all the pigment is gone. The algae is gone. You're just seeing this transparent coral polyp. And what you're actually seeing is the coral skeleton, because the coral has no color anymore, no pigment anymore. Now, if the water temperature returns to normal and the intensity of the sun kind of goes down a bit, the coral can reabsorb the algae and go, everything is good. We survived. We bleached, but we survived. But if this goes on for too long, the coral will eventually starve to death because it's lost its capacity to produce nutrients. It doesn't get enough nutrients anymore. So in essence, it's a stress response that the coral has. It goes, I'm a bit stressed, I'm really uncomfortable, you're making me even more uncomfortable, let's get rid of you to try to sort of calm things down here. But it can also happen when other things in the water chemistry changes. So that when you've got like a flood, freshwater flood, for example, happens, all of a sudden you salt water, the salinity drops really quickly, and the corals will get stressed again, and they will also expel the algae. So it's a stress response, more or less.
Yeah, so when we talk about Cyclone Narelle before, obviously there's the impact on the coral reef of the storm itself. But if we're seeing a lot of flooding along the coastal areas and the water being pushed back out into sea, that can also have an impact on the corals. Is that right?
Yeah, it can. I guess with the cyclones, a lot of the water that comes out is also full of sediments. So they can also be inundated and get just a lot of sediments landing on them, which means that the photosynthesis doesn't work anymore because there's no sunlight coming to the algae. So all of these things, like it's lack of sunlight, it's too much sunlight. So yeah, they are a bit fickle. They love things to just be the way they want them to be.
So, which brings us to making corals stronger, I guess. So globally, we're seeing coral restoration efforts increasingly focus on strengthening corals' natural heat tolerance. What does that mean in practice?
Well, I mean, in practice, it means we're trying to figure out what makes some corals deal with heat stress or stress better than others. What's behind that kind of capacity to withstand it? Are some of them just more tolerant to that uptick in photosynthesis and go, we don't really mind, you guys can have a bit of a sort of haywire photosynthesis event going on here, but I don't really mind that much? Or are some of the algae better at dealing with this high light and temperature, and they don't go into that photosynthesis overdrive? Or does the coral get less stress if it's in really good condition overall? You know, if we have had good sleep, good food, we're feeling very fit, then stress doesn't affect us as much. So maybe just the coral being in better condition is a good thing to do. Does it make a difference if the coral's been exposed to heat stress before and sort of built up some tolerance? You can even look at ways in which the coral can survive for longer without the algae. I mean, corals do also feed plankton in the water. Some corals do that better than others. So you can also look at what lies behind this capacity for a coral to eat and get nutrients in other ways than from their algae. I mean, we've been working on this for quite a long time at the foundation. Like I did say in my introduction, I was part of the very, very early work on this, looking at the genetics behind heat tolerance. I did that. That was over 20 years ago now, close to 20 years ago now. And that was actually funded by the Great Barrier Reef Foundation back in the days. So we've been doing this and funding this and seeing the importance of this for a very, very long time. And I mean, back then we didn't have anywhere near the genetic tools that we have now. You need a lot of the genetic tools. You need to understand what it is that triggers this, if it can be inherited, if it's a conditioning thing, or if it's a kind of they're born that way. And another major program that the Great Barrier Reef Foundation funded back in the early days was something called the Reef Futures Genomics Program, where we really started putting some of the very first complete coral genomes together to give us that critical foundational knowledge about what underpins so much of the work that is currently happening. We were also, as many of you might know, partners of the Reef Restoration and Adaptation Program, and brought a lot of those partners together. And that brought together Australian and global expertise in coral adaptation and made a significant progress over the last 5 to 6 years. So we've really seen a lot of progress in the last 5 to 6 years to help solve this challenge. And we are still funding a lot of this critical research in this field, especially in the symbiosis field right now, because that's where we're seeing the most progress.
So can you talk about that a little bit more, like these early signs or these signs that the work that we are doing is working?
Yeah, so like I said, the most promising work that we're seeing, or where we're really seeing some significant progress, is in the algal symbiont. They have very, very short generation times, which means that you can breed them to increase their heat tolerance quite quickly in the lab. You can ramp up the tolerance. The more tolerant ones reproduce, and after a few generations, you've got a batch of Symbiodinium, or these little algae that are much more tolerant to heat, that don't get as stressed in the heat events. So we're doing that, and we've got some of these heat tolerant strains in the lab. We have inoculated coral, like little coral polyps, with them. We know that the corals that are inoculated with the more heat tolerant strains do withstand heat stress much, much better. And we kind of are starting to look at ways to put these out on the reefs and see if they maintain that connection with that algae. Other things that we've seen is conditioning dust matter. I mentioned that thing that healthy corals seem to withstand heat stress much better. So feeding coral larvae during restoration kind of things with certain plankton during development gives them a much better chance of survival in the field if they get stressed. So condition does matter. Previous thermal stress events do build level of protection as well. So we do see the mass bleaching event that happened over in WA recently was in part caused by the fact that those corals had never experienced severe heat stress before. So they were quite naive, and when this hit, they all kind of suffered a lot worse than what they would have had if they'd had some incremental heat stress provided sort of on them earlier. Some other cool things, we know that corals that live in extreme environments, like in mangroves, for example, that get really sort of hot, they also have a lot of plankton and nutrients in the water. They seem to be much better at feeding on plankton. So when they bleach, they survive for longer because they're actually better at heterotrophic feeding, as we call it, like at eating plankton that is in the water. What have we else done? We have seen success in breeding trials. You can breed 2 heat-tolerant parents and get a more heat-tolerant baby coral out of that. However, we can't predict when it works and when it doesn't work. Some heat-tolerant parents don't produce a heat-tolerant baby, and we don't quite know the genetics behind it yet. So there's still a lot of work trying to unlock the genetic codes that will give us more certainty in these selective breeding things. But yeah, sometimes parents are just more heat tolerant because they have been acclimatized, and that doesn't mean that their babies will be. So unlocking that genetic background so that we can be very selective in how we breed them and what parents we use.
Yeah, so lots of really exciting new research, new innovations that you're seeing. How far are we from applying these solutions at a meaningful scale?
So, I mean, we can do, like I said, we can do things like selective breeding of heat-tolerant parents. We just don't have a 100% success rate at the other end of it. But I mean, that's okay. You can still kind of make active choices about what parents you use in breeding, not in breeding experience, in breeding for coral restoration programs. So it is a really good start. It means that we can choose what parents to cross. It means that we can go out and make sure that the corals that have survived bleaching events are brought back into the lab and are used in these big sort of selective breeding programs. The challenge is, of course, to get it right all the time. And then some of the other challenges that we do have is, like I said, you can put the algae in there, you can put those corals out, and they do show higher tolerance to sort of high stress events. But how do they maintain that connection with that particular algae? Or how do we apply it to corals in the field so we don't have to take it in and inoculate little larvae? So it's that treatments at scale is still a major challenge. Like, how do you get these treatments out there at the scale that we need them to?
And are we looking at that as well? So looking at not just, I mean, when we spoke to Mary before, looking at lowering the cost, making sure that we're doing things so we can go, we can look at both the testing and refining, but also looking at that future pathway to scale. While we're looking at the research that you've mentioned, are we also looking at what that next step is?
Yeah, absolutely. So, I mean, 2 things in that. One thing actually links back to some of the things that Mary was talking about with this eDNA thing. Like, we can actually tag corals that we have bred for heat tolerance in the lab, or that we have treated with symbionts and so on, with DNA tags. And there's researchers at the Australian Institute of Marine Science that are currently doing this, going, how do we insert the genetic tags into them? That means that 5 years from now, in a major bleaching event, we can go out and sample the water and go, oh, that coral actually is still here. It's still alive. It did survive. It did actually infer heat tolerance that we can still monitor and manage and look at, so sort of out several years after we've put them out in the field. So those kind of innovations are really, really cool to be able to follow these corals and see how long they survive out there. What other, I mean, we are also really looking into that deployment at scale, like treatment at scale out in the water, looking at things like releasing little lipid balls containing probiotic, like healthy microbes to put on corals out there, looking at using underwater robots to deploy treatments and so on. But I mean, a lot of that is still early stages, and a lot of it is still like concept in people's minds. But yes, absolutely, it's going to be the next big challenge, and I think it's going to be something that we will really want to look into and solve. It's a challenge that needs solving, absolutely.
Yeah, and a last question before we'll get into some audience questions afterwards, but We talk a lot, you know, about the Great Barrier Reef and the work that we're doing here, but what does all of this innovation and this research mean for the potential to support reefs around the world?
Yeah, no, I mean, there is a lot of interest to a lot of this new technology and a lot of this new research. And of course, this research is happening elsewhere in the world. It's not just happening on the Great Barrier Reef. We have great partners with people working in the Red Sea, for example, which is an extremely hot ocean where corals are surviving temperatures that are much, much higher than what we're seeing here on the Great Barrier Reef. So having those like global collaboration and global partnerships is really important. And we are about to start a big global program where we're going to pull together a panel of the best scientists in the world looking at this, what we're calling assisted adaptation challenge together, and trying to unlock that and trying to set up parallel experiments here and in the Red Sea to see what the difference is sort of on a geographical scale between the same species and so on. The Great Barrier Reef Foundation is also currently working with a program with some partners in the Pacific, where we are really trying to democratize some of this technology and see what it will look like in other areas outside the Great Barrier Reef and how that will work, and partnering with some excellent scientists over there as well.
Brilliant. Thank you, Petra. That was amazing. Lots happening across both in Australia and globally. And so we might move on to, I know there's quite a few, I was just having a quick look, there's quite a few questions. Welcome back, Mary. So, I mean, we've covered a lot of ground today, from detecting crown-of-thorns, starfish outbreaks earlier to strengthening corals for the future. So, well, I'll have a little look and see here. We'll go through some of these questions, and please pop all your questions into the chat. For those we don't get to, I guess I'll do a shameless plug for our Great Barrier Reef Foundation newsletter here. We'll put the link in a bit later. If you do sign up to this newsletter, we'll make sure we answer all of the questions in the next edition as well. So we will cover them all. First one to Mary. So is there a risk to culling native species in terms of ecosystem balance?
Yeah, that's a really great question, and it's one we think really deeply about. So I think the first thing to know is the goal of control isn't eradication, because these are a native species. It's really about bringing numbers back into balance when they outbreak. So part of our innovation program, our team develops the really sophisticated models that guide the control teams in the targets they use for culling. So it's about bringing numbers back into ecological sustainability. So starfish numbers stay at levels where their impact of their feeding doesn't cause coral decline. So that's really important. Ecological balance, not eradication. These starfish have a right to exist. We just don't want them destroying the habitat of all the other the animals on the reef.
Brilliant. And I'll go on to the next one because it's another COTS question, Mary. How do we control explosive COTS breeding after bleaching events?
Okay. Well, yeah, I saw that one in the chat, and it also talked about culling robots and things. So I don't know that there is evidence of explosive breeding after bleaching events, but certainly these two pressures can compound on the reef. And right now, we have teams of divers who obviously are really targeted in what they where and when they take action, and increasingly have innovations and tools to help them make those decisions. And one of the things we're working on developing is robots and artificial intelligence to help determine where those outbreaks are happening, where the starfish are starting to emerge. And that's not so much to get robots to do the culling themselves, but to do that surveillance for us. So that's another key area of innovation for our program.
Brilliant, thank you. And Petra, we'll throw to you for this next one. What temperatures can corals survive in? What's the maximum temperature? And I might just read the next, there's a comment for the next that somebody has written, Reefs of Hope in Fiji is currently moving heat-tolerant corals from hot pockets to cooler water in order to save them at this time. Currently, we're seeing 34 to 36 degrees Celsius temperatures. So just to let you know that somebody has kind of responded to that from a different perspective.
Yeah, and I mean, of course, that is a very good question. I mentioned briefly this thing working in the Red Sea where corals just withstand much, much higher temperatures because that's what they're used to. So the quick answer is they can deal with 2 or 3 degrees above what their natural high maximum temperatures are. And they vary. Like corals in the southern Great Barrier Reef are much less overall heat tolerant than corals in the northern Great Barrier Reef, if you're looking at temperatures exactly. So it depends on what they're used to. But they are, as I also mentioned, quite fickle. They're used to, they live in the tropics where temperatures are quite stable. The water quality is really good. So their total temperature variation is not great, but I'd say most of them about 3 degrees over what they're used to being their maximum degree, maximum sort of water temperature, they're going to start getting quite uncomfortable. And then it's also how long it stays that warm matters as well.
Yeah, I guess a follow-up question from me, not from the audience, but is there a time period, like an average time period that, you know, if they do go up those few extra degrees in the warming water temperature, that when do we need to see it come back down for those corals to survive? Is there any sort of average that you see there?
I mean, we are really trying to aim, we're working with something called degree heating weeks, which is literally the accumulated heat stress over a certain period of time. So as you can see by that term, like you can have an accumulated heat thing happening for a few weeks, and some of them will last for 1 week, and some of them, there was recently a paper that came out from it was over in WA, some of the southern reefs in WA, and they found corals there that could withstand 20 to 30 degree heating weeks all of a sudden, like unheard of before. So that too varies a lot. Some of them are more sensitive than others. I would say in general, the more stable the temperature is where the coral lives natively, the quicker it gets stressed. The more it's used to temperature fluctuations happening, the longer it will probably withstand a bit of stress. But as we start looking into this and start doing a lot of experiments, there's so much variation out there that it is really, really difficult to give a specific answer. You're just trying to improve them overall, just trying to make them incrementally a bit better at withstanding the heat stress.
Yeah, thank you. Question for both of you, Mary, we'll start with you. Are there any other countries already starting to use the type of innovations that you've spoken about today?
Well, here in Australia, we're actually really at the forefront in terms of using eDNA monitoring for starfish outbreaks, at least crown-of-thorns starfish outbreaks. And so that's because it's really only becoming operationally ready right now. So we're really on the forefront of this. But now that we have this capability, there's absolutely a great opportunity to start sharing that experience, especially with other countries who routinely get outbreaks, Japan, the Philippines, elsewhere in the Pacific.
And Petra?
Yes, there are absolutely other countries doing a lot of this work. And as I said, we do work a lot with countries in the Red Sea that are quite advanced in many of these research areas and methods as well. But I'd say one of the things that we might not think about or might not realize, in Palau, there is a research group from the University of Newcastle in the UK that are working with the Palau International Coral Reef Centre that have one of the longest standing intergenerational breeding for heat tolerance programs there is. They have like a families of corals that have been tagged for generations there. So they have a really, really good handle on which parents are actually heat tolerant and infer that heat tolerance to their offspring. Not necessarily why or what genetic markers there are, but one of the most advanced experiments in selective breeding is happening in this tiny, tiny little island up in Micronesia in Palau, which is super exciting because we are now lucky to work with them in our program there in the Reef Seeding Initiative.
Yeah. Only got a few minutes left, but Mary, I might ask you, that's been put in here. I've been reading about passive eDNA samplers. Do these have any potential for continuously monitoring COTS hotspots?
Yeah, I mean, that's a really good question. I think the key thing we're looking at here, so passive samplers that technology won't enable us to get good quantitative information on eDNA. And so really, that's probably not the right solution for this particular problem. Those passive samplers are great for lots of different other applications, but in this case, we need that, that quantitative information because we expect, like I said, we expect COTS to be on the reef, so we expect to be detecting them a lot. It's about how many there are.
Brilliant. And I've just got a little timer on my phone telling me we've just got 2 minutes left for the Q&A. So I might do one more question to both of you. I have to say, for everybody there who are reading in the chat, there are a lot of questions and a lot of very scientific questions. And again, just will point out that we will put the answers in our next newsletter. So for those of you who have put questions in, we really do appreciate it, and we will get to them. But maybe for Petra, what are the main coral species that you're working with in your heat resistance research? And what species are showing to have higher tolerance to heat stress and which species are not doing as well?
What we're working with, if you take the example from Palau, for example, it's a lot of acroprid corals, a lot of plating corals. The primary reason behind that is they are corals that are really, really important for the for corals to come back after a devastating kind of bleaching impact or crown-of-thorns outbreak. These are the early colonizers, so having them back on the reef is important. They also have a reproductive system that makes it quite easy to do selective breeding because they broadcast spawn. They broadcast spawn at times when we can predict that they do it and so on. So a lot of the early work has been in the acroprid species out there. With regards to withstanding heat stress better than others, I mean, we have seen that these branching, fast-growing acroprids, poeciloprids, they do bleach really quickly. They also come back really quickly. The massive corals are a bit more tolerant, the big, bolder, the slow-growing ones, but once they die, they are really back. So difficult to get back into sort of recolonize a reef with. So yes, they might be more tolerant, but they're also more susceptible to being sort of they're more susceptible to being lost. Once they get bleached, once they die, they're really hard to get back, would be what I say.
Brilliant. Thank you, Petra. And last question for Mary. Mary, is there any way, there's obviously animals that do eat crown-of-thorns starfish. So is there any way to protect and increase the numbers of those animals to help with this problem as well?
Yeah, that is such a great question, and we have a whole certainly some really brilliant scientists looking into that that exact question. So what we know is that we actually used to think these starfish only really had one predator or two because, my goodness, they do not look very appetizing, right? But they actually have plenty of predators, and we're learning that all the time. We're actually using eDNA to find to prove that there are signals of these starfish in the guts of some of the fish species that we actually like to fish for. So what we find is that in areas that are protected from fishing, so green zones on the Great Barrier Reef, we have much lower incidence of these starfish outbreaks. And that just goes to show that having those networks, fishing in the right places really is something that helps protect the reef from starfish outbreaks. So things like spangled emperor are actually one of the starfish's predators, especially when they're in juvenile phase. So those are one of those species that the more we protect and make sure we have those green zones and we we all do our bit to only fish in the right zones, that really helps the cause.
Brilliant. Thank you. And thanks both Petra and Mary for both answering my questions and then answering the ones from those of you who have popped them in the chat. And I think, I mean, what we've seen today, what we've heard today is that there are these challenges facing the reef, and they are significant, but there are solutions that are underway that are being worked on right now, and they're working. They're being trialled, they're being tested, and we're looking at ways to use them across not just more of the Great Barrier Reef, but reefs around the world. But scaling this work does depend on continued support. So if you would like to help drive the science and the innovation that will power the future of coral reef protection and innovation, please head to our website where you can read more about the work that we are doing. And if you are in a position to do so, there's also ability to make a donation. Every donation helps to take this work further and faster, and together we can close the gap between ambition and action. So again, I want to thank Mary and Petra for joining us, for taking us through the work that you are doing, and also the work that you're doing with many other people within the reef protection and restoration space, both here in Australia and around the world. Thank you, everybody, for joining us from all of us here at the Great Barrier Reef Foundation. Thank you, and have a wonderful day.